and then did postgraduate training at the world-famous Cook County Hospital. I mean, that must have been an experience. And he started his academic career at the University of Pittsburgh Medical Center, then moved to Northwestern University, and then ended up down the street, or yeah, literally south, you know, at the University of Chicago, where he is an associate professor, as I mentioned, in the section of general surgery. The unique part of the story is that, you know, you're a surgeon and you work in intensivists. And to have that coupled with computational biology and complex systems work is really a very attractive and interesting combination. Gary has been a leader in the swarm community for years and years. He knows several folks at our Center for the Study of Complex Systems. Carl Simonson regrets for not being here, but he's with you in spirit. You study, you know, wound healing, host pathogen interactions, and all kinds of stuff, and uses computational approaches and simulation and modeling, and is a consumer of supercomputing cycles. You know, at Livermore, at the Oak Ridge National Laboratory, probably with Jaguar, some of those systems, and you know, years and years ago, we used to do a lot of supercomputing when we were in the center, and, you know, we don't do so much of it. I think Zhang does some. But so it's going to be really interesting to see and to learn from Gary how simulation and modeling approaches, forward approaches, and I think I saw Dan here, you know, are really, you know, part of the repertoire. And what comes after big data, that is a really good question. And so we're actually, especially with the launch of our Data Science Institute, you know, and separating the hype from reality in big data, I think is one of our challenges. And the role of dynamic knowledge representation in addressing the translational dilemma in the supercomputing age. That's an excellent title, and we're looking forward to hearing you elaborate on that. Gary, welcome. We're really happy that you're here.
Thank you very much. Thanks, Brian, for having me. Thanks, Kevin, for having me as well. You know, as Brian mentioned, I am a clinician. That's my only area of formal training. So I live in blessed ignorance of mathematics and computer science and actually basic biomedical research. So to a certain degree, that allowed me to be a relative blank slate going forward when I started my research career, but will also give me an excuse in case I state some clear falsehoods. As a disclosure, I'm a consultant for a company called Immunetrics in Pittsburgh that does large-scale in silico trials of sepsis and rheumatoid arthritis. So given my background as a clinician, my primary goal is to try and take care of people, right? I want to fix people. And when I was a young trauma surgeon at Cook County Hospital in the early 90s, I spent my energies on making sure I was the best surgeon, best intensivist I could be, and yet I found it somehow wanting. And it turns out that I was not the only person, because the FDA put out this critical path document back in 2004, which I'm sure all of you have seen, where on the screen left, you can see expenditures on biomedical research, and on screen right, the number of deliverables to the bedside. And as I mentioned, I'm not a math person, but those two graphs should not be going in opposite directions, right? There's a problem with respect to this discrepancy, and it only appears to be getting worse. And in the last decade, that has not gotten any better, right? So we have this paradoxical situation where we know more and more about the biological systems we presume to want to be able to control and treat, and yet our tools to be able to do that are wanting. So I apologize for the next slide, but as a clinical surgeon, I have to show at least one gross surgical picture. So this is someone who I took care of about probably about 5 or 6 years ago now, an unfortunate young man who got stabbed at the base of his right neck and his right chest, lacerated his internal mammary artery through his innominate vein, bled into his chest, required about 70 units of blood in the first 24 hours in order to control this. For those of you who don't know, the human body holds about 10 to 12 units of blood. And during the course of this procedure, despite how dramatic this looks, so the guy's head is over there, his feet are over here, this is his chest, which is open, there's his lung sitting on his chest, Because when a patient bleeds that much, when you give them that fluid back, they swell up. They develop what's called abdominal compartment syndrome. So we just basically have to cut the abdomen open and sew a plastic bag on top of it so the pressure can be relieved to allow the other vital organs to work. This is a technical exercise. This is trivial. This is cutting and sewing. Right now, surgeons spend a lot of time learning how to do this well, but the fact of the matter is this is just woodworking. It's carpentry work. Okay, and the fact of the matter is that when I'm in the operating room doing this at 1 o'clock in the morning, and sure, the patient's immediate survival depends on how well I happen to do those particular technical exercises. While I'm doing this, I know that the primary determinant of whether or not this patient is going to survive their hospitalization is whether or not they are going to get over the inevitable multisystem organ failure, need for dialysis, nosocomial pneumonias, ventilator-associated pneumonias, ventilator-induced injury, and all the potential problems associated with their medical care up until that particular time where that patient may be fortunate enough to leave. And the fact of the matter is, even though we have some idea about what the molecular processes are involved in that, and I don't need any sort of test to predict that those things are going to happen, I can't do anything about them before they happen. I can give antibiotics. I can support the lungs as best I can. I can put the guy on dialysis. But as far as making him better, causing his biological system to increase its trajectory back towards a state of health, I have not a single tool. And nobody does. Right? We can't address what's actually wrong with these particular people to make them better. So that's very frustrating, and that's what prompted my interest in this, because I have some idea what's going on, but I can't do anything about it. So that led me to a diagnostic approach to what may be the gaps in our current tool development pipeline in taking care of patients. So I'm a first principles kind of guy. What we're actually interested in all medicine is organ function, right? Because disease is a dysfunction of some organ at some level. Right, so we want to be able to fix this. Now we know organs are composed of multiple individual cells, which are themselves composed of multiple interacting molecular pathways. And what we're asking pharma to do in this day and age is, given the complexity of this particular system, infer ahead of time what potentially manipulating this molecule is going to have on that entire system. And I propose that we can't do that intuitively. We can do this potentially augmented with computational tools for data analysis and simulation, but doing this intuitively does not work. It does not work because biological systems are organized in such a way that there are essentially epistemological boundaries between the multiple hierarchies of organization, such that you can't necessarily intuit what the effect of manipulating at one level of organization is going to be at the next level of organization, right? Things do not necessarily commute across these levels of organization, right? So this is a problem. This is a particular issue because this is what our biomedical experimental workflow is, where each one of these particular sets represents an experimental platform, and we are making inferences with respect to what we gather, information that we gather from one platform, and trying to translate it up this particular organizational scale, right? And the question is, what is the justification for the knowledge that we are punitively translating, right? We do it intuitively. This particular model system has this particular pathway. Ergo, it must behave in a similar fashion when you move to another platform. Is that valid? Can we test that in some sort of way? And that leads to the question is, what is similar, what is conserved when you move up this translational investigatory pipeline? So as an example, a tale of two papers, this infamous paper from the GLUE grant a couple years ago that was in PNAS and in the New York Times, right? Which led to much gnashing of teeth and rending of hair and all sorts of stuff associated with how these people should be burned at the stake, and led to response papers like this, okay? Which, these are intentionally provocative. Now, so let's leave aside for a second that there might actually be some methodological concerns with either of these two particular approaches. The fact of the matter is that both of these groups are respected scientific groups. They both utilize tools that they interpret using their expertise, and they come to completely different results. And it becomes then a political issue, right, as to which one you're actually going to believe. This, however, does not help you getting towards a solution. So the question then is, how do we actually affect this translational goal by taking basic science knowledge and moving it up the organizational pipeline to get to clinical output? So I would pose that this slide represents our current operational strategy for doing this. Okay. We hope for the best. We hope that we've guessed that this particular preclinical platform works. We hope that this particular drug candidate is going to survive phase 1, 2, and 3 trials and not kill people in phase 4, right? We hope that these things are going to happen. And I would propose that this is not a very robust strategy, right? We should be able to do better than this. We should be able to utilize our knowledge of what the scientific process is and the advances in technology to be able to show our work, take this miracle component out of it, and actually make this in an explicit fashion such that, and this is important, our failures are usable, right? What we want to be able to have is usable failure, because if we're counting on a miracle that doesn't happen, then we don't really know much more than what we had to begin with. Right. So, as I mentioned, we have had a revolution in science where big data is powerful, right? Data center, right? And this is incredibly important, revolutionized how we look at systems and how we analyze and can interpret what might actually be going on in these very complex systems that we're dealing with. Right? This guy, Nate Silver, he's a University of Chicago person, being able to predict elections with a certain amount of precision. But he cannot tell you how to win an election, right? He can tell you who's going to win. He cannot tell you what you're going to need to do in order to win that election, right? So if we want to, in biomedical research, in the biomedical endeavor, do something other than just describe what might be happening, we need to move beyond what data can provide, which is extremely powerful. But it's a trap, okay? It's possible that if you just get stuck in the idea that if you can collect enough information and throw a big enough compute at it, that you can get answers. And that's just another form of a miracle, right? Because you got to move beyond what that big data can actually provide to be able to do something, to be able to fix patients, right? So first principles kind of guy. Science is science, right? This is a scientific cycle. We all know this. Francis Bacon, Isaac Newton, 16th century kind of stuff. Observations and data, identification of patterns, interpretation of that data, construction of hypotheses, testing of hypotheses with experiment. This has worked for 500 plus years. Our sum total of technological advance is based on this particular cycle. And yet I'm telling you, it is not working as well as it should work in biomedical research at the moment. And the question is why? Well, on one hand, part of it has to do with the complexity of biological systems and the diseases that we're trying to deal with today. Right? So those particular diseases that are linearly reducible to a single cause, right? So in a very coarse sense, if you take the appendix out in appendicitis, you don't have appendicitis anymore. If you decide that you should stop drinking your own feces and water, you're not going to get cholera anymore, right? So public health in certain aspects of surgery, are linearly reducible. But when we're dealing with wound healing, cancer, AIDS, aging, diabetes, obesity, these are diseases that represent dysfunctions of systems where they're shifted, where there may not be a single cause that can be reduced out in this particular cycle. The other problem is that we have an embarrassment of riches with the increase in the dimensionality of the data that we can collect. So much so that our scientific cycle is imbalanced. So technology has allowed us to be able to gather more and more information out of each particular biological system, and advanced computational tools are able to extract that information out of those particular experiments, and we're actually able to make inferences out of those particular patterns into potential hypotheses. But this critical step of testing hypotheses to check the plausibility of our punitive evaluations of causality, right? This remains in a serial form, reliant upon the life cycles of cells and rats and the labor of graduate students and postdocs, right? We cannot keep up, so there's a bottleneck here. So from a diagnostic standpoint, as a clinician, this is where we want to target this particular problem, right? So thinking about science in a particular way, right? This is another projection of this, right? You have data that you do all the great stuff you do with great data, and you generate hypotheses, but they remain hypotheses. They're possible explanations for what you have. What's needed, is to close that loop with some sort of means of evaluating the causal mechanisms that are inferred from that particular data analysis. Okay? This is a cyclical process, obviously. And the emphasis here is on data. Knowledge. Interpreted data. Data that has somehow been processed, evaluated, looked at, and comes out with something that says, I think this is what this data means. And the question is, how do we test that particular process? Now, part of the problem is that hypotheses are always incomplete, because you can't know everything about everything as much as we would like to know. And therefore, at some level, if we want to be able to get to useful information, our goal should be sufficiency of explanation. And I'll get back to this in a second. So one of my favorite authors has this little thing about the well, so model is a very much of a loaded term. It means very different things to different people. A model is essentially a formal representation of one system with another system, right? So you can have a statistical model, you can have a conceptual model, you can have a model that is a computer program, etc. All right, I'm going to make a distinction between what I'm talking about in terms of simulations or executable dynamic models, right? And that is a means of closing that loop, because you can have a data model of a data structure and come up with a hypothesis, right? But turning that hypothesis and making it go, right, is the role of a simulation or an executable model. Right, so this is Borges, this little short story on exactitude in science, right? You've had time to read this essentially a little bit. But this is a trap, right? Because on one hand, we kind of think we want something that's exactly like what we have, because that's precise. On the other hand, you end up with something that's as complex as the thing you started with, and then you can't really do anything more with that. So here's a little example of this. This is Marcus Covert did this a couple years ago at Stanford, where they simulated the simplest bacterium that you possibly could do with a cluster of 128 computers, right? So only 525 genes, did 10 hours to do one cell division, which is actually longer than the biology, and generate this amount of data, which is a impressive technological engineering feat. But the fact of the matter is, the goal of science is selective abstraction, right? Because if everything is its own precise thing, then how can you compare it to another thing, which has got its own complexity and its own identity? What we want to be able to know is what level of abstraction of description of one particular system is applicable to another level of system, right? And if we think about the quantitative sciences with their production of theory, right, abstraction's a pathway to theory. The idea of finding natural laws or commonalities that have greater explanatory power, right? And that's actually what we want to have, is we want to have knowledge that can explain as much as possible, right? So this is a little bit different than how people may think about how you gain increased information about a system, right? As opposed to degree of detail of knowledge which is important, but how can that knowledge be translated from one system to another? So biology carries its own challenges, right? I showed you the organizational component, and it has this legacy of being almost completely empirical. I mean, biology has one theory, right, evolution, and that's pretty much it, right? And therefore, the tendency is to focus on increased degree of description, which gives us this problem of infinite regress. So again, first principles guy. So let's go back and say, what are the questions of science and how they fit into this particular paradigm, right? So what are we studying? How does that system work? And why does that system exist in that particular way, right? And the idea is that you want to gain deeper scientific knowledge that has more general applicability through greater explanatory power, right? And this is the key towards understanding how biological systems work. Right, so what is the what of biology? Well, this is description, right? All the data concerning a particular system goes into describing a phenotype of that particular system, and it represents a description, right? And this is agnostic to scale, because you can have metadata, population data, heredity data, all the way down to your different levels of omics. So here's a fanciful classification system for biological systems, right, again from Borges. So this may seem kind of funny or ridiculous, but on the other hand, this is agnostic at scale. And there's no reason that this is an invalid means of describing systems or biological objects, right? It's as valid as anything else. You can do. Yeah, there you go. So you can have different classifiers based on these particular components. Right. This is the zoological or botany basis legacy of biology, right? Biology up until the time of Darwin and Mendel and molecular biology was basically a descriptive. But we want to know what the how of biology works. And why is that important? Because it gets us to function. Function is what we actually view in terms of this, what disease actually represents, which is dysfunction. You need to know function before you can characterize dysfunction. And this is the means by which one phenotype transitions to another phenotype, right? It is what goes in between. It is the dynamics of how biological systems exist. All right, so it's a dynamic interpretation of data, right? How does one data set transform into another data set in a biological sense? And scale becomes important, right? Because as I showed you with that nested hierarchy of organization, events don't necessarily commute, right? So scale is actually important. And then finally, the why, right? So generally, people don't like to talk about the why because the Aristotelian final cause issue, which leads to teleology and intelligent design and that kind of stuff. But in actuality, right, we do have a final cause in biology, which is evolution, right? Because if we know one thing about biological systems is that every single biological system we have is a product of evolution. And therefore, that particular process represents a constraint on how these things can actually emerge, right? So evolution is diversity plus fitness, selection, and persistence. And we'll come back to this concept a little bit. So modeling the simulation in bioscience, I think about as a path towards theories, binding together disparate systems and disparate data sets. And because of that, right, because of the uncertainties associated with biology, right, we have to be able to have some plausible validity first before we get into precise mapping between one model system to another model. Right? This plausibility is very important, right? Because it's not an engineering task where you have a constraint system, you have relatively comprehensive knowledge of how the components of that constraint system works, and you can do optimization, right? Which is not biology because we don't know what the rules are. We don't know what those structures are. Right? So building robust models along the lines of a model simulation is akin to building robust explanatory theories, little t theories, right? But the key here is you need to rapidly iterate this with experimentalists who are the experts in actually extracting knowledge out of biological systems. So a colleague of mine came up with this idea of translational systems biology, more words, 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 right? Because all the other words have been taken. This, of course, is nice because it's all translational, right? It's systems and it's biology. But, right? But what sets this apart, we think, is that the focus is on constructing clinical phenotypes, right? So we're not modeling, as important as they are, yeast, bacteria per se, individual experimental platforms, et cetera, which again are incredibly important in gaining knowledge about mechanism. But our goal is to affect that translational step to take that knowledge and contextualize it in something that's clinically relevant. And this is important because control is the goal, right? As a physician, it is not adequate for me to be able to diagnose and prognose. I need to be able to do something about it. So just as my patient who was stabbed in the neck, right? I don't need any tests to tell me what he's going to get. Doesn't matter, though. I can't do anything about it, right, ahead of time. I have to wait for it to happen. So in order to turn applied science into actionable knowledge, we need to know punitive mechanisms. We need to have inference of causality, because those are the things that our interventions, our focuses of control are going to happen on. Right, so we build these simulation models with the idea of investigating control. Right, so we talked a little bit about the importance of dynamics. The idea here is that these are evolving systems. They shift over time. They are actually very robust, because even with your disease states, most of the processes are actually functioning in a programmed sort of way. But this is important, right? Because there is no disease modeling without health modeling. You can't model pathophysiology without knowing the physiology from whence the pathophysiology arose, because then you don't know how you get back to the healthy physiology, which is what the goal is. So therefore, every model I'm going to show you starts off as a model of a healthy system that, if you perturb it in a particular way, becomes a disease state. And therefore, we consider disease as a specific dynamic state. So as Brian mentioned, I do agent-based modeling. I'll run through this very quickly, but agent-based modeling is a discrete event, object-oriented, rule-based computer simulation method that basically creates virtual objects that operate on series of rules. You create virtual populations, and you let them run around, and you construct essentially in silico experimental platforms that you can now use like other experimental platforms. Right. The keys here are you're generating population behavior, and it encapsulates biological stochasticity, whether that's true stochasticity at the gene regulatory level or stochasticity that's due to measurement unknowability. This is important because anyone who's ever worked with a cell culture can realize that you can have monoclonal cells, perturb that in one particular way, and you get a distribution of how those cells act. Okay, and that's a fact. So if you take the slide number 3, so every bioscience talk has slide number 3. 1 is the title, 2 is the theory or hypothesis, 3 is the diagram, right? That shows all the arrows and stuff like that. If you take that diagram, you add its stochastics into it, you bed them in computational agents, you can essentially try and reconstruct what a biological system may act like. So most people know agent-based modeling through popular culture. In these movies, this is a screenshot from Lord of the Rings. These battle scenes, like the zombies and World War Z, et cetera, these are all agent-based models, right? Where each individual creature is a computer model sitting on a checkerboard or a hex board and has certain rules associated with, I'm facing this direction. If I have X number of orcs in front of me, fight. If I have Y number of elves behind me, stand and fight or run away or whatever. Okay, and the idea is that this mimics how many biological systems work, and therefore you get these very biomorphic sorts of appearing behaviors. However, right, so each creature is an individual program. When they started doing these simulations for these CGIs, what they found is that they couldn't get battles to occur, because they programmed what were actually very plausible stand and fight rules. But once you put them in a population that incorporated space and neighborhoods, right? Every time the armies came together, they turned around and ran away, right? Because they couldn't anticipate that population level effects could be inferred by how individuals behave because they couldn't do it. Right? So the solution was to turn the survival instinct way down, which there's a lesson in there somewhere. I don't know. But the one I choose to take is that there's power in population. Right? That there's the ability to encapsulate the range of behaviors that individual cells, for want of a better term, can take. So the idea is that you can now use agent-based models to cross this translational divide, right, where the reductionist necessary, absolutely necessary reductionist mechanism by which we do basic research, by taking things apart and finding out how things actually work, we can reverse Humpty Dumpty and reconstruct that particular system using an agent-based model in some form of abstraction and plausibility. So I'm going to give you 3 examples of where this works, right, or my examples of how I've used it. I don't know whether you can tell me whether you think it works or not. Right. So the first one is actually my first work because as a critical care surgeon, my interest was in sepsis, and I started the thing that actually prompted me to get interested in this was the failure of the initial anti-cytokine trials, anti-TNF, anti-IL-1, anti-endotoxin. Right? It led to, again, existential crisis in the sepsis community. What's going wrong? Blah, 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 and then retrenchment. Right? But what I took out of this is, well, maybe if you had a model that represented the knowledge that people had when they made those clinical trials and you used it, maybe it would have told you that it didn't work. It wasn't going to work. Right? So this is a old model, right? It's over almost 15 years old now. And basically, it's a representation of the state-of-the-art thinking about how systemic inflammation worked in the 1990s at the time that the anti-cytokine trials were done. Cell user cell types, mediators, whatever, relatively abstract. So as I mentioned, I'm not a computer science kind of person. So I use a computer programming platform called NetLogo, which is intended to teach K through 12 students about ants and traffic and stuff like that. That's my level of computing programming expertise. But what you see here is an endothelial surface that's perturbed with a trauma. And you see inflammatory cells responding to that, secondary injuries, et cetera, et cetera. There's stochastics involved here. Basically, it's an abstracted endothelial blood interface that I use as a proxy for an individual. Right? Again, abstraction. Now, despite its level of abstraction, it is plausible because it is able to reproduce all 4 big buckets in behavior to injury. Right? It can either heal, it can have pro-inflammatory SIRS and die, it can have immune-compromised multi-organ failure where it's susceptible to an otherwise trivial insult and then die, or it can have overwhelming injury. Again, very large abstract buckets. It actually is valid because it reproduces the cytokine profiles associated with clinical patients who have sepsis. Right? So every legal rule that's now put in that particular model can then be manipulated as an example of a control mechanism. So what I did was I took that particular model and I simulated these anti-cytokine trials, both the ones that were failed and some ones that were in animal studies, and combination therapies which were not logistically able to be done. And the question is, given the knowledge that you have and a representation that behaves plausibly, right, do any of these things actually change outcome? And the fact is, they do not. Right? So the critique of this paper, of course, is that we already knew what the answer was. None of those things work. Okay? But the fact of the matter is that the process by which this was done assumed that they should have worked, right? Because I gave the drugs every chance to do exactly what they were intended to do, right? So no question about bioavailability, no question about whether the dosing was appropriate. Those things are fixed, right? So given a system described in this fashion and you perturb it in that particular way, it doesn't work, right? So the system wasn't so robust it couldn't be changed because if you gave antibiotics, the things got better, which is, again, plausible. Right, so watching this gave us insight because what happened was if you gave the intervention, the TNF would go away for the time the anti-TNF was administered, but the damage is still there. right? It shuts the entire system down such that the system can't heal, so it just delays what the input is. So you get this pebble in the stream effect, right, associated with the intervention, right? And the problem was not parallel pathway redundancy, which is one of the things that were raised at that point in time, like you didn't block enough of the stuff. It's temporal robustness, because the system can't correct because you're blocking the things that are needed for the thing to actually correct, right? Which, subsequent to this, is realized that sepsis is not endogenous anti-TNF monoclonal antibody deficiency, right? That's not what the disease is. It's a different thing, right? And therefore, that particular intervention is not the appropriate thing to do. And systems died because they couldn't clear the initial damage and recover quickly enough, right? And this is what we actually see. Right, so the functionality of this is essentially, you can think of that computer simulation as an instantiated thought experiment, right, where it's a conceptual model, it's a hypothesis that you can now visualize and see how it can actually go. And you advance by taking hypotheses that don't behave plausibly and throwing them away, and advance plausible ones. And this is something that can be done at Canada Discovery. This paper was a decade old. Abyssinia is a pan-European initiative to look at in silico trials. I'm going to their next meeting in Barcelona. Some of you may be familiar with this, right? But this is coming down the pipe. Right, so now I'm going to talk a little bit about cancer. I'm not a cancer biologist. Again, I know nothing about these sorts of things. I had a student who come to me and said, I want to do a project in breast cancer. Like, okay, that's fine. So how does breast maintain its own duct epithelial population at baseline? What's the healthy state? So we decided to start with that. Right, and this is the schematic that shows the components of this. Right, so it requires all these other than duct cell pathways. And when we put this into a NetLogo program, it is plausible because it reproduces the cycling of the breast tissue associated with hormonal stimulation, and actually shows expansion of breast cells in pregnancy. And it also is calibrated to hormonal cycles with respect to proliferation of breast tissue. So the basic cell population levels are plausible. And then we said, okay, now we're going to add oncogenesis. And what is oncogenesis? Well, oncogenesis is DNA damage, introduction of mutations. So we created a synthetic genome that has particular functions that, if they get knocked out, no longer work in that particular model, right? And these are our functional gene groups. Right? So the other, 9,992 genes, they don't do anything, right? Because we don't care about that. These are genes that are associated with oncogenesis. If they get knocked out, then the things that they're supposed to do don't happen anymore. Right? So what we then did is we wanted to now bridge this mechanism to population level modeling. So we simulated 40 years of menstrual cycles. So this is premenopausal cancer, right? And now, since cancer is a rare event and takes a long time to develop, we define cancer as unregulated growth and tried to see whether or not we can match the incidence of development of cancers over time. So the big problem is with that particular conceptual model, you couldn't get any ER positive cancers, which is a problem. If you go back really quick, our only way for cancers to propagate is if their damaged cells replicate and ER positive cells don't replicate. So we said, well, how do we get ER positive cells to replicate? Right, so CMET's involved in suppression of their replication. So the question is, what suppresses CMET in an ER-positive cell? And our student found this thing called RUNX3, which is a tumor suppressor gene that's identified in gastric cancers. And we put that into the particular model, and we generated plausible population dynamics of both wild-type and BRCA knockouts, both in terms of endpoints and trajectories with respect to seer population data. Right? And RUNX3, I think, has also been shown to have the increase expressed in ER-positive tumors on a ex post facto evaluation. So this also led us to think about a more in-depth question about what cancer is. Because again, I don't know anything about cancer. I don't know how all these different pathways, et cetera. Right? But first principles approach, what is cancer? Well, first of all, cancer only occurs in multicellular organisms. Bacteria don't get cancer, right? And they're disordered cells with disordered growth. So the question then is, what are the failure points that are possible from an evolutionary standpoint that lead to cancer behavior, which looks like unicellular behavior? So came up with this generative hierarchy for cancer, right, where first order processes represent actually the DNA damage to the base pair switches or the production of SNP. Second order processes are the manifestation of those particular disorders or dysfunctions as cellular dysfunction, so problems associated with apoptosis, loss of growth regulation, etc. And then third order processes, which are how do those cells behave within the context of the tissue, right? So these are stromal effects, angiogenesis, etc. Now, the source of the damage, the cause of the DNA, we always know that they're external things, toxins, radiation, etc. But inflammation, of course, is a well-known intrinsic biological process that's associated with this. So the question is now, can we contextualize cancer in an inflammatory milieu? We come up with this schematic, right, where you have an inflammatory damage, these different kinds of processes. And the key here is that you're shifting these damaged cells, first and second order disordered cells, into now colony behavior, so they behave like unicellular organisms, where, from an evolutionary standpoint, genetic plasticity is not a bug, it's a feature, right? Because unicellular organisms utilize genetic plasticity to survive. So now you have a cancer that is utilizing its plasticity of its genome, its ability to increase its incidence of mutations, as a means to escape any sort of therapy that you want to do that's predicated upon eradication, right? Because that's what evolution is, right? To fight eradication. Same sort of synthetic genome. And what this shows, basically, as you would kind of expect, it cannot an unplausible output that as you increase the amount of inflammation, you generate increased number of cancers and these microtumors. And that increased cellular disorder is associated with more inflammation, which again, this came out about a year before, but that paper in Nature about the number of the cancerous chance type of thing, right? Where the more replications you actually have, the more chance you have mutations, which seems so obvious, but somehow it's not. Right, but the here is that the disorder leads to increased possibility of resistance to any particular therapy. So the implications for cancer treatment are that you can't account for second or third order processes unless you deal with the first order process, because then you're just skimming the top of the iceberg off, right? So eradicative therapies that induce inflammation are bound to generate resistance, right, by the very nature of how that system is structured. So what you want to work for is potential environmental control as opposed to eradicative strategies. Take those components of the cells that are actually functioning, right, and use them to make them be happy, right? So you live with the tumor as a follower. That gets us to host-pathogen interaction, right? So this is a big schematic. This is a very hot topic right now, microbiome, all that kind of stuff. All right, so I'm going to give you a couple examples of how we've looked at host-microbe interactions primarily in the gut. So there's this disease called necrotizing enterocolitis, right? Greatest cause of neonatal gastrointestinal morbidity and mortality. And basically what happens is if you have premature babies and if you feed them, sometimes their gut turns black and dies. And there's a bacterial component, but there's not a specific bacteria that's associated with this particular disease. It's hard to study because in order to do this, they need to like throw little baby rats into hypoxia chambers or refrigerators and stuff like that, which we don't do with children. So the question is whether or not the experimental thing that you're using to generate a particular phenotype is actually mimicking the processes that generate the particular disease. So we came up with a minimally sufficient hypothesis that immaturity of reactive oxygen species management in immature enterocytes allowed them to become fragile as a result of metabolic stress, which a mature enterocyte doesn't have. So the usual diagram is all the stuff that goes into the agent-based model. And what this model does is it generates the different disease spaces associated with necrotizing enterocolitis, where you can have a subclinical survival phenotype, this patchy neck scare phenotype, which is important clinically, right, because that's your actionable window, and then full thickness necrosis, which you have to operate on. And the idea here is that you can generate a disease space based on the fragility of the enterocytes and the virulence potential of their microbiome, not tied to a specific bacteria, but what that virulence potential is within the patient's microbiome at itself. So that might be manipulated with formula feeds, maternal milk, other types of probiotic agents, etc. But it gives you potential control knobs that you can now triangulate in a space to reduce risk. So those used abstract bacteria. This is an example of more detailed bacteria. I collaborate with a guy, John Alverdy, at the University of Chicago. Right, he studies Pseudomonas virulence and gut-derived sepsis. So what we did was we took an abstract model of the gut ecology, and we embedded models with his rules. So this is exactly dynamic knowledge representation of that diagram, which is the canonical diagram that represents what his lab does. So if I go to a particular basic science researcher and I say, what do you know? They write this big thing on a whiteboard and say, okay, I can put that into this program. We'll see how that runs. And what we do with this particular model is we run it along with his in vivo experiments to help try and predict and examine what the various states are in between his sampling points or in conditions that he can't logistically do with his particular rats. Last model I'm going to talk to you about is a spatially explicit all-purpose model of enteric tissue. We really struggled to get something that would come out to a word. This is a virtual gut that basically recapitulates the histology of the gut surface. And this is our current state-of-the-art gut model. And it has this control structure with the idea of reproducing histology. Histology is important because that's still used as a marker for disease clinically. Right, so these are the pathways incorporated. Orange is inflammation, blue is morphogenesis. And as with the RUNX3 situation, we need to make a hypothesis as to what a potential link was between these two particular pathways. We came up with P10, which has subsequently been demonstrated to be actually upregulated in a particular disease state that I'll talk about in a second. Right, but the model maps to the histology. So here's the gut, crypt villi. We construct a set of rectangular boxes that have grid spaces on them. This is the grid space unfolded in the model. Right? And this is on a single processor. You let this run. But if you do the supercomputing version of it, you can generate essentially a villus architecture here that looks like the villus architecture of the gut. And so this is a supercomputing implementation, right, where each one of these little dots is an agent. So this model is validated because it generates plausible mediator gradients with respect to the morphogens. And it's general purpose because it doesn't have one disease that it replicates, because it replicates a state of health. Right? It's able to reproduce healing of local injury. It's able to reproduce the effects of ischemia reperfusion on the gut mucosa. And it simulates metaplastic change as would be seen in the pouch of a patient who had remedial surgery for ulcerative colitis. Right? By changing the crypt-villus ratio due to persistent low-grade inflammatory stimuli. So we've extended this particular model, as I mentioned, to segment HPC, which is a supercomputing implementation, to C++, geometric partitioning for those of you who are computer science interested. We've run it on a Cray, the Beagle at U of C, which has 12,000 cores. We can simulate about 60 square centimeters of tissue. This is the paper in PLOS One just came out. And the idea is that now we can scale across molecular scale to anatomic scale issues like flow dynamics, tissue architecture, etc. Right, so we have a director's grant on this particular project and a BD2K R01 pending. Hopefully we'll get funded. Yep, and we have allocations on Sierra at Livermore, Mira at Argonne, and Titan at Oak Ridge, which is our largest machines to run this. This is the UO1 that we have just sent in, the MSM consortium, right, that uses segment to study pouchitis. Again, hopefully, we'll see how this goes. So, in this, the microbiome is relatively abstracted, but it represents a control feature, and this goes into a Department of Energy initiative called BASIC, right? So I'll just talk very briefly about this. The BASIC initiative is an attempt from these national labs to look for something for their supercomputers to do because they're tired of simulating nuclear bombs, right? So they want to be able to use their petascale, exascale computers to simulate something else. So they're looking at biology to do that. Right, so it's a consortium of federal DOE labs, academic partners, and industry from tech, Intel, Amazon, blah, blah, blah. Right, and the idea is to get direct federal earmarks along the sides of the exascale computing initiative, means a couple $2 billion a year to do this. Right, so the current basic track structure has predictive pharmacology, pathophysiology, and pathobiome. This is done at Harvard by Joe Lascazo, who's the chair of medicine at the Brigham, I believe. Tim Buckman at Emory is running the sepsis predictive pathway. I'm leading the modeling component of that particular pathway. And I'm also co-lead of the predictive pathobiome, which is looking at antimicrobial resistance potential in the microbiomes of ICU patients. Right, so I'm sorry, I'm going through a lot of this stuff fast. We can talk in more detail about these things at my talk tomorrow. Right. So I'm here to be recruited. Not going to tell you anything different. Right. So these are the rolling horizon of deliverables that we want to try and do. Right. So there are short-term things, classical bioengineering, things that can give us clinically actionable output right now. Then there are medium-term data-centric things, prognosis, diagnosis, disease classification, bio-knowledge extraction. But to actually get to the endpoint of being able to engineer new therapies and really deliver personalized precision medicine, you're going to have to go to simulation. Right? It has to be this way, right? Because the key is being able to represent outliers, right? And simulations allow you the possibility of being able to explain outliers. So when you do data, too many possibilities, right? We all know this. Increased dimensionality of data, increased dimensionality of explanation, right? And theories are the things that constrain what those explanations are, right? And this is where the personalization is. Right? Generation of state from those particular constraints. Right? So this is our translational goal. And as much as we would like there to be, there is no shortcut to that scientific method. Right? We still have to test. We still have to evaluate what those hypotheses actually are. Right? But we can potentially accelerate that cycle. So this is the last thing I'm going to show you. We have come up, my colleagues and I, with this thing called the computational modeling assistant. Right? Where you essentially semi-automate those processes that can be automated in the construction of computational models. Right, so what this does is it basically a big database integrator where you take knowledge that's expressed in near natural language form and it uses a logical rewrite system to say, if I have a hypothesis of this particular structure, you can generate computational simulation models of these particular types. And it will generate code for that, at least first set of code, and will tell you how those simulation experiments should be run. Right? And what it does is it takes, to a certain degree, the modeling and simulation computational biologist a little bit out of the loop, because we all know how difficult it is to actually get that ball rolling. Right? It is a collaboration facilitator. For those of you who are interested, right, this is what the software infrastructure for the CMA actually is. Right? It uses COCA. This is the data level SQL system. Application tier uses MAUD, which is a logical rewrite system from U of I Urbana-Champaign. and the simulation engine that's based in BioCoca, BioSwarm, runs on GPUs, but can be used on other clusters, right? The guy, I don't know anything about how this actually works. This guy, Scott Christley, is the computer scientist who works with me, who developed this system alone. So what does this all get you? Right, so if you have a model that behaves in a plausible fashion, well, then that's plausible. And now you look for ways to break it, because that's how science advances, right? Falsification. And if it doesn't match, then you say, okay, now I'm going to go back and look at that hypothesis structure and say, what parts of those things I am not sure about, right? And since electrons are cheap, you can run all those different possibilities and see which ones actually behave plausibly. So you always have to remember this Mark Twain quote, it ain't what you don't know that gets you into trouble, it's what you know for sure that just ain't so. Right? So you always go back to first principles and check what your underlying hypotheses are. The vision for the future is that scientific research community lives in knowledge ecologies, right, where we use evolutionary principles on science, where you have all the wonderful data collection and interpretation capabilities placed into a HPC augmented hypothesis testing environment that allows people to investigate, sort out, and select those hypothesis structures that pass the test of new observables, right? Right. This is the fitness function. Those hypotheses that are fit because they can match newly observed data are the things that persist. And this cycle goes on. This is what I talked about in this 2010 paper about bridging causality and correlation in the petaflop age. It requires these types of platforms. By the way, I wrote a book about this, Translational Systems Biology. Pretty expensive. All right, that's it. Any questions? Sorry, it's a lot of stuff, but. Right.
Yeah. Oh, I have to use a microphone. I can yell too. So that's good, but I go back to that Mark Twain quote. With these agent-based models, you're assuming that you know sort of what the knowledge base is. And the reality is, for example, in the central nervous system in humans, we don't really know a lot of the pathways and what's going on there. So any kind of agent-based model would have to have some kind of built-in ability to be, I don't know, accept new data and extract it from that knowledge to make the agents accurate.
Right. So the question is, how do you deal with unknowability?
Right. Which is a big problem.
Yeah. So how do we deal with it now? We just say, oh, I don't know that. I try and get some more information. Right. The question is, we don't know what we know. Right. So assuming that this is incomplete, assuming that it might be wrong, right? Assuming all those sorts of things, this gives you a visualization of how that actually might behave. I would say that that's actually a very useful piece of information because you go into it knowing it doesn't represent ontological truth. You go into it knowing that there are going to be gaps. Just as I demonstrated in our examples, our models were wrong with respect to the breast cancer and with respect to the initial link between morphogenesis and inflammation. The question then is it now focuses you on finding what is the thing that's actually missing, right? So, of course they're incomplete. They're going to always be incomplete. The question is whether or not the model you have is good enough to represent enough of the data that's out there, such that that model could be potentially useful in engineering that, some therapy or prediction for that particular system. Yeah. 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 Well, this follows along with the knowledge that becomes acquired. Right, so part of the problem with the Kuhnian paradigm is you don't know it's happened until after it's gone. Right, he doesn't tell you how to bring it about. Right, you can't, I mean, by definition, right, you can't do anything about that. Right, so, yeah, that's right, you are in the midst of it. So the question then is whether or not you can utilize any sort of tools that will allow you to gain an understanding into what you actually know now. Right? So explanatory models like the type you're talking about, right, represent the endpoint of that is still a hypothesis structure. The degree of precision with respect to you believe the hypothesis is up to you and your trust with the particular system. But the fact is that now you want to manipulate that particular system. Right? If you only use an inference program, you can't test that until after you've collected the data on that whatever intervention you've actually done, because you can't test that. Right? What we want to be able to do is project forward into what are the possible sets that might happen, understanding that it's not going to be perfect. Right? This is why it's still an open research area. Right? We want to be able to develop the tools to increase our level of trust in those particular projecting simulations to allow us to move forward.
Okay, good. Liz.
Yeah, so it strikes me, again, along these lines that, your model is based on putting in data that simulates a model, and you're testing sort of the integrity of what you're putting in. But again, to extend that to things that are outside of that model, I just don't see how your system can do that. And maybe I'm just missing something.
Right. So one way to think about what these models are is they're essentially hypothesis representations for conceptual model verification. Right? So using verification in a computer science sense as whether or not your conceptual model behaves the way you think it's going to behave, right? Given all the multiple interconnections that are present. Right? That's the first test. Because if it doesn't behave the way you think it behaves, then either there's something wrong with the model, which is possible, right? How that model was implemented, but there might be something unanticipated in what you actually thought your knowledge actually represented.
Right, but I think you're trying to sort of sell a bigger vision that I'm not sure I'm seeing your methods able to do, right? So right now, science tests something very, I guess, microscopically in a dish, right? You have very controlled experiments, very controlled outcomes. And then there's like the population reality, which is a macro environment, which is very different than a microscopic environment. And really, you're trying to sell that you want to make that leap. You want to be able to make these big predictive models in the population, but really you're using data from that micro environment.
Well, so you're using some of that data, and you are not constructing a model of that data. You are constructing a representation of the generative mechanisms that produce that particular data. Right? So it's very important in my mind to separate the data, which is the list of observables that you have that are your, let's use Plato for a second, the shadows on the wall, right, represented by the light that you can see, but you don't know what's behind, right, that's casting that shadow. You make inferences about that, okay? So what this does is it says, okay, I have this shadow that looks this particular way, and this shadow that looks this particular way, and I know they're somehow kind of similar. Is there some sort of conserved generative mechanism that can allow you to represent both of those? Now, my argument is that this is a question of parameters as opposed to model structure. So let me use the thing I talked about this morning with Dan, is that, I think it was Kayvon, where the range of biological behaviors in human people, in many ways, is an issue of parameters, not necessarily components, because we all kind of have the same genes, a little here, a little there, right? But even an individual, because I'm sick now, my parameter set is different than it was when I wasn't sick. Okay, but it's the same model structure underneath. So what you want to be able to do is to have a conserved enough, robust model structure such that its regions of parameter space can capture outliers. And this is the key. It's the importance of outliers. So if you construct a data model, and I don't know enough about how data is done, right? But my understanding is that if you have a very wide data set, right, you want to be able to try and find the best fit, right? You don't necessarily, and by doing that, you have to a certain degree, accept that your fit is not going to hit those outliers. Now, the fact is, those outliers were generated, assuming it's not, experimental error or whatever, right? The biological system had the capability to generate those particular configurations, however it happened to be able to do that. There is some fundamental level of representation of those generative mechanisms that can allow you to do that in a simulation, right? So this BD2K grant that I'm having, right, kind of flips the idea about what it means to parameterize a simulation. So of course, in systems biology, the parameterization of your model is a big deal, as you would expect it to be, right? And unfortunately, many of them are too brittle because the parameters are relatively small. Okay? That's actually not a very explanatory model. That's a very precise model, but it's not very explanatory. Right? What you want to be able to do is to have a broader exploration of what model parameter space is and say, can I actually reproduce these outliers with this particular model structure with those particular sets of parameters? Now what you're looking is at as categorization of parameter values when you evaluate biological systems as opposed to specific component types. Does that, you don't buy it? You don't buy it. I mean, it's it's how physics works.
Kevin.
It's how chemistry. It's how
Yeah. Any model that the more variables you put into, let's say, a linear regression, the more predictive it is. But then you overfit at the end of the day.
well, there you go.
Your data set,
Right.
and then it doesn't.
Yeah, so that's
and
a difference between
generalizability goes down, right? So I mean, I understand the concept, but yeah, I still don't see. The limitation of this is the data that you're putting in, which is coming from hardcore experiments, right? Which I think you're.
But it's not the data you're putting in. It's the knowledge that you are using to construct how that data is generated. Right. But that's what we operate on.
Yeah. I just want to make a comment. I mean, as a clinician, I find the agent-based modeling sort of exciting because it's providing a sort of a more phenotypic output, which is really important. And the other thing is that it's probabilistic as opposed to deterministic. And so when we run these models time and time again, I mean, we'll get an output plus or minus a standard deviation, which is, again, what we observe biologically. But I don't think Dr. Ann is suggesting this model as opposed to all others. I think it's an important tool. And when you get to multi-scale modeling, there are certain aspects of biology that are best modeled using certain techniques. And this is certainly, I think, one of those. So again, when you're looking at really complex systems, inflammation and coagulation and those sorts of things, where concentrations and neighborhoods become really important, this gets really, really exciting, in my opinion.
Right. The CMA that we've developed doesn't just build agent-based models. It will build any kind of computational model that you have, that we have specification.
Dan?
Just a simple question. You're emphasizing somehow the value and role of the outliers. And in these simulations, just a methodological question, is it possible to go in and pull out the data points that correspond to the outliers and run the mechanism backwards to see what it was? Like you say, within the biological potential of the system, you can generate this outlying behavior. So can you actually do an etiology of your outlier? Why did it go bad?
Yeah, that's what we want to be able to do, right? Because I currently do not have the resources to do that degree of path investigation, right? So this is the model checking that was invented at CMU, I think, with Ed. I can't remember his last name. But this is the idea, right? Is that you take the ones that are your unusual ones and you backtrace those and see whether or not how it got to that particular point is a plausible sort of thing. I think outliers are important because that's where the interesting biology is. So that the capacity to generate outliers is the key to why we have heterogeneous populations, differential responses to disease, and why we need to have something like this to get to precision medicine. The precision medicine is just the right drug for a right person, right? It's the right drug for a right person at a particular time based on their particular health state at that particular.
Gary, you, in an earlier slide, you defined evolution and gave some kind of sub-steps in the process of evolution. Are you utilizing those principles kind of as an expansion of the genetic algorithm in some of your simulation efforts?
I have not. I have not done that because when I talk to certain computer scientists, these models have too much randomness already, right? And GAs, you know, are black boxes, and we don't actually know what's going on with respect to that. We are looking at the use of GAs and other types of machine learning to build potential classifiers for the behavior spaces of these particular models and see. I mean, came on, I had a great conversation this morning about a potential project where we might be able to do this reverse machine engineering to map back and forth between simulated data sets and potential clinical.
Mm-hmm.
You have like input or parameter or anything that you're dealing with, but when it goes to the predictions, you have predictions that works and the ones that don't work, like outliers. Reverse engineering, reverse machine learning allows you to train another related model to trace this back to the parameters. Basically improve the
related to what Dan was saying to a certain degree.
It goes back and forth through a process to make sure that your model is actually, instead of overfitting, it basically matches the reality of the data.
Mm-hmm. Thank you. Okay, any last questions? Well, I think you've gone through Plato and, you know, Francis Bacon. I mean, you took us through over 2,000 years of thinking about science. And Thomas Kuhn jumped in there towards the end. So lots of good ideas and lots of practice. So thank you very much. It's very
Appreciate
stimulating.
Your time and your questions.
Yep.